All right. So, uh, up to this point, we have talked about glycolysis, and to spatially situate us, glycolysis is happening in the cytoplasm. The uh, enzymes required for each of the steps of glycolysis are to produce pyruvate, to produce ATP, the 2-ATP, and to produce our reducing equivalents are occurring in the cytoplasm. Our lecture today is going to take us from the cytoplasm into the mitochondria. And so, well, the two processes that we're going to be focusing on today are going to be the TCA cycle and oxidative phosphorylation. And so these two steps are happening within the mitochondria, but in different spatial locations of the mitochondria. The TCA cycle is occurring within, you can think of it as the almost like the cytoplasm of the mitochondria. We call that the mitochondrial matrix. That's where the TCA cycle is taking place. And then oxidative phosphorylation is going to be happening on the membrane of our mitochondria. And so our mitochondria have a unique membrane. It's this double-walled membrane. It's a double membrane structure with an outer membrane and an inner membrane. Once the slide comes back, you'll be able to see that the inner membrane possesses these transporters and pumps. There we go. These transporters and pumps that are really key for oxidative phosphorylation. And there's this space between these two membranes called the intermembrane space, where you can see on this slide, there's going to be this accumulation of hydrogen ions. These hydrogen ions are going to be really key for a final step of producing ATP at the end of oxidative phosphorylation. What's key about these processes that you'll be able to appreciate is that there is this ability to produce far more ATP than what we typically, with what we get compared to glycolysis alone. So what we're going to talk about today is going to enable our cells to produce far more ATP when we take pyruvate that's produced at the end of glycolysis, when we only have a couple of ATP to produce much more. And so again, today we'll be situated within the mitochondria here, talking about the TCA cycle and oxidative phosphorylation. These two steps are dependent on the presence of oxygen here. And so this is going to be all about aerobic respiration, requiring oxygen, and occurring primarily within eukaryotic cells. And so this aerobic respiration is not going to be taking place amongst prokaryotic cells. Again, the two major steps we're going to be focusing on are the TCA cycle and oxidative phosphorylation, both abbreviated to the TCA cycle or OXPHOS. The TCA standing for tricarboxylic acid. What that comes from is that there's going to be 3 carbon dioxides produced during the TCA cycle. It's also called the Krebs cycle. The scientist, Dr. Krebs, was the one who first characterized this cycle, also known as the citric acid cycle, because throughout the course of this process, there's going to be different variations of citric acid that make up some of the intermediates. And so whenever you hear the names citric acid cycle, Krebs cycle, TCA cycle, they're all talking about the same process, just trying to name it based on different components. A couple of key roles about the TCA cycle is going to be that it's going to be generating more reducing equivalents, and these reducing equivalents are going to be key for feeding into OXPHOS. And second, what's really key is that it's going to be an entry point for amino acids into our metabolism. So a lot of times when there's a period of starvation or fasting, muscle wasting will occur, breaking down muscles into their constitutive amino acid parts and using those amino acids to feed into the TCA cycle. And so that's how protein can be used to feed into metabolism as an energy source. That's also going to be true not just of protein, but we're also going to be talking at the very end about how lipids can be used as an energy source as well. For oxidative phosphorylation, another name for it is the electron transport chain. And so again, when you hear oxidative phosphorylation or electron transport chain, those two things are largely talking about the same thing, just focusing on naming it based on different aspects. A key part of OXPHOS is going to be creating this proton gradient. We saw on the previous slide, in the intermembrane space, this accumulation of these hydrogen ions there. This proton gradient that's pumping hydrogen ions into that space are going to be really key for a series of coenzymes that are really important for oxidative phosphorylation. And we're going to be using that gradient. So it's mediated by these coenzymes, but then that proton gradient is really crucial because those hydrogen ions will be used by an ATP synthase enzyme. ATP synthase, it's an enzyme that synthesizes ATP. It uses hydrogen ions to start producing ATP. And so these are the kind of like the big picture, what are the roles of each of these processes, key things to keep in mind to help us as we start to dive a little deeper into each of these processes. And so these will kind of help you keep your bearings as we kind of get into the more granular details. So this is a overall summary slide of the TCA cycle here. At the very top left of this image, we have our pyruvate that's produced from glycolysis. We talked last class of what happens immediately after that pyruvate in the presence of oxygen, where pyruvate dehydrogenase takes the pyruvate and produces acetyl-CoA. And there's a reducing equivalent produced from there, and our very first carbon dioxide molecule. That acetyl-CoA is going to feed into the TCA cycle here. What's important to note is that there's going to be a series of reducing equivalents produced, series of carbon dioxide molecules produced, 3 of them again. And what can sometimes help as we're keeping track of things is taking note of the carbon backbone for each of these molecules. They can be drawn structurally different ways, but drawing them in this format can actually be really helpful to try and keep track of being able to appreciate how these molecules are broken down. And so we have, get my mouse here. So we have this 3-carbon structure here with pyruvate. We lose a carbon dioxide, so now we have a 2-carbon backbone here. That makes up the acetyl chain, the acetyl group, and then we have coenzyme A. So we have acetyl-CoA here that then is going to feed into the TCA cycle. We have oxaloacetate, which has a couple of carboxyl groups on 2 ends, and we're going to have a series of reactions that are going to produce citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then all the way back to oxaloacetate. I'm not going to ask you to memorize each of these intermediates. There's going to be a key couple intermediates that I'm going to point out to you throughout the course of this lecture because of how they feed into other information that's going to help layer on top of this. But the key things to take note of are the production of carbon dioxide molecules, the production of these reducing equivalents, and the production of a new reducing equivalent that we haven't talked about yet, FADH2, that's going to be produced at the 6th step here. And so what's key to note is that the coenzyme A that's attached to acetyl-CoA, it helps shuttle these acetyl groups that are used and leveraged throughout the TCA cycle. And so if we look at the structure of this molecule, it's going to look somewhat familiar to a couple of other things we've looked at before. We have an adenine and a ribose sugar. Together, those make up adenosine. We have this pyrophosphate bridge and pantothenic acid. What's key to know is that pantothenic acid is actually vitamin B5. And so there's a series of instances during metabolism where a component of some large structure is going to be one of the molecules that's also dubbed a vitamin. And so that's going to happen a few times, and I'll make sure to point it out, but it's also a reminder to take your multivitamins if you can. And so it's going to, the combination of coenzyme A with the acetyl group provides the entry point into aerobic respiration, into the TCA cycle. Acetyl-CoA is also going to be really, really important for the beta oxidation of fatty acids. And so it's going to help, acetyl-CoA is going to be really key for the ability to take lipids, to take fatty acids, and use them as an energy substrate where from lipids, they can undergo an oxidation event that produces acetyl-CoA that then feeds into the TCA cycle. Or in reverse, we can go from acetyl-CoA that's produced as a result from glycolysis with our pyruvate that becomes acetyl-CoA via pyruvate dehydrogenase. And if there's an abundance of acetyl-CoA, it can then be stored or it can then be catalyzed enzymatically to become fatty acids and lead to fat accumulation or fat synthesis, I should say. So again, our main entry point is the production of acetyl-CoA from pyruvate that produces our reducing equivalent in the first carbon dioxide in the TCA cycle or tricarboxylic acid cycle. And so key takeaway from the entry cycle is we get a reducing equivalent and we get acetyl-CoA that lets us enter the TCA cycle. Acetyl-CoA donates two of its carbons to oxaloacetate to produce citrate via hydrolysis event. And so the carbon backbone is highlighted one color so you can appreciate how many carbons there are in the backbone, as well as the carboxyl groups are highlighted in different color to help you appreciate how many carboxyl groups, and you can appreciate how oxaloacetate and acetyl-CoA have come together here to produce citrate. The subsequent steps will produce a oxidation event that'll produce our first another reducing equivalent here when we go from when we produce alpha ketoglutarate as well as producing succinyl-CoA. So we have two more reducing equivalents that are produced here. And so when we go from succinyl-CoA to succinate, what's important to note is that we have our first ATP or it can be GTP that are produced. There are enzymes that mediate this reaction going from succinyl-CoA to succinate. Depending on the species of that enzyme will determine whether an ATP or a GTP is produced. But the key thing to note is that we do have a energy substrate that is produced during the TCA cycle. And so again, if we think about starting from one glucose molecule to getting to this step here, we had two pyruvates that are produced from one glucose. So it's important to note that we have two ATP, or it can be two GTP, but for simplicity's sake, when we're taking inventory of how many ATP molecules are produced, I'm just going to refer to these as ATP here. So we have from one glucose, two ATP that are produced here during the TCA cycle. And so we have two more carbon dioxide molecules that are also produced. So we had one carbon dioxide produced when we're generating acetyl-CoA, and two more down here during these steps to produce alpha ketoglutarate and succinyl-CoA. Pronunciating these intermediates is going to require me to speak a little slower. So forgive me if I mispronounce things a couple times. And so the key thing to know is that we have our three carbon dioxides that have been produced here. Finally, we have our new reducing equivalent species that's also been produced here. We've only been talking about NADH. We have another species of reducing equivalent called FADH2. And so FAD accepts the electron transfer during this oxidation event when we produce fumarate. And so we have a single, during one TCA cycle, one FADH2 that's produced. And finally, another oxidation event that produces again, we go back to oxaloacetate and the production of another reducing equivalent. And so when we get back to oxaloacetate, it's ready to accept another acetyl-CoA to start the TCA cycle back up and continuing to produce these reducing equivalents as well as that ATP molecule. With FADH2, again, I should say with FAD, we have a somewhat similar structure as what we were just talking about before with coenzyme A. We have an adenine and ribose, which makes adenosine here, a pyrophosphate bridge, and the oxidized form of riboflavin has a box turned around on the left here. Riboflavin, I believe, is vitamin B2. And so again, we have another instance where our intermediates make up one of the vitamin B complexes or complex of B vitamins that are important for metabolism. And so we have the oxidized form of riboflavin to the right here. And so the ability to have this other species of reducing equivalent is going to be important when we get to oxidative phosphorylation. So the TCA cycle in total from one acetyl-CoA that feeds into the cycle, we're gonna produce three. I should say this is per acetyl-CoA, three NADH reducing equivalents, one FADH2, and one ATP. And so again, for each glucose, that reaction happens twice because during aerobic respiration, per glucose, we're going to produce two acetyl-CoAs. So our net yield so far, if we include what comes from glycolysis, what comes from that entry point step to produce acetyl-CoA, that entry point step to produce acetyl-CoA, and then the TCA cycle itself, we're going to have 4 ATP, 2 from glycolysis, 2 from the TCA cycle. We're going to have 10 NADH. We're going to have 2 NADHs from glycolysis itself. We're going to have, and then 6 generated from the TCA cycle, and 2 more NADHs from the entry step to produce acetyl-CoA. And we're going to have 2 FADH2 produced during the TCA cycle. And so what's going to be key is that we're going to, the next step of oxidative phosphorylation is going to use these reducing equivalents, NADH and FADH2, to feed into those processes. And these have received electrons that have been released during oxidation steps throughout glycolysis, throughout the TCA cycle. I'll stop here and give space for any questions. All right, if there's no questions, we can press on, but if any questions pop up, please feel free to raise your hand in the middle of the slide. So now that we've produced our reducing equivalents, NADH and FADH2, what's going to end up happening is rather than receiving the electrons during an oxidation event, they are now going to be the ones during an oxidation event losing the electrons and having them go into this electron transport chain or this electron transport system on the inner membrane of our mitochondria. There's a series of proteins on the inner membrane of our mitochondria that leverage these electrons to start creating this proton gradient of hydrogen ions that will then lead to the production of ATP. And so both NADH and FADH2 were going to be losing their electrons during an oxidation event. And what we can see here is, as an example, during the activity of a specific dehydrogenase, we're going to have NADH2 losing electrons that go into this first protein that's going to be complex 1. We have complex 1, complex 3, and complex 4 here, as indicated by the Roman numerals on the protein. And during this oxidation event, we're also going to have these hydrogen ions moving across complex 1, complex 3, and complex 4 into the inner membrane space. So what happens first at complex 1, NADH is going to lose its electrons, and we're going to have the hydrogen ions, 4 hydrogen ions moving across complex 1. It's going to be important to remember how many hydrogen ions are moving across these complexes. There's only going to be 4 of them total, but complex 1 will have 4 hydrogen ions moving across, and the electrons will then be passed on to these intermediate proteins, as well as iron sulfate. We also have these coenzymes, coenzyme Q, and these cytochrome proteins that are going to be passing these electrons across from one another across the inner membrane to then get to a point where we're going to produce a couple of molecules at the end of the electron transport chain. But the key thing to note is that we have 4 hydrogen ions pass across complex 1 here. During the generation of coenzyme Q, we're going to have 2 more hydrogen ions moved across here, 2 hydrogen ions moved across complex 3, and 2 hydrogen ions moved across complex 4. And so as this, as the electrons are moved from each of these molecules from FMN to iron sulfate to coenzyme Q to cytochrome proteins, those electrons are going to carry a charge with them. They're going to change the conformation of each of these complex proteins. That'll then open up these complex proteins to enable hydrogen to move across the inner membrane into that inner membrane space. That then is going to culminate in the ATP synthase pump to drive 3 hydrogen ions down across it and produce 1 ATP every time it does that. And so the I was going to say recent, but now it's been 12 years, so it's weird to. There was a paper published in Nature back 12 years ago that did a really deep analysis and characterization of these proton pumps, of these electron transport complex proteins that demonstrate whenever we have the oxidation event of these electrons being donated across those different molecules I pointed out in the previous slide, they were able to demonstrate and illustrate the actual opening and closing that enables hydrogen ions to move across the inner membrane. And so every time we have the oxidation event, it opens up the bottom, and when there's a reduction event, it opens up the top. And so there's this back and forth of opening and closing of these protein complexes on the mitochondrial membrane that enables hydrogen ions to move across them. And again, the ability for these hydrogen ions to get into that inner membrane space is critical, is necessary for that ATP synthase, that ATP pump to generate ATP. It's dependent on these hydrogen ions being there. And so for those hydrogen ions to be there, it's these electron transport protein complexes that enable that to happen. And again, the main thing I want you to note is that the transfer of those electrons confer a conformational change that opens and closes those complexes. Yes. Yeah, so the oxidation event where NADH has lost its electrons now, and those electrons are now present in the intermediate carriers, whether it's I think it's flavin mononucleotide, iron sulfate, coenzyme Q, the cytochrome proteins. As those electrons are passed across those like intermediate carriers, depending on where we are in those complex 1, complex 3, complex 4, that will those electrons they carry a charge. By nature of carrying charge, they're going to change the conformation of those complex proteins. And so once they're in their reduced state, they're going to open up at the top. When they're in their oxidized state, they're going to open up at the bottom. And so there's this like back and forth of receiving the hydrogen ions and passing them into the inner membrane space. Does that help answer the question? Yeah. Yeah. And so this is happening for this is happening for these all 3 of these protein complexes on the membrane of the mitochondria. Complex 1, complex 3, complex 4. We have a complex 2. We're going to get to that in a second. But the key thing to note again is that we have this passing over of electrons, the driving of hydrogen ions into the inner membrane space. 4 at complex 1. We have, if you combine what happens with coenzyme Q, we have 4 at complex 3 and only 2 at complex 4. And the final destination of those water molecules is this generation of water from hydrogen ions and oxygen here. And so at the end of this passing through of the electrons across that membrane here in these protein complexes, we have the generation of water. And again, the last 2 hydrogen ions that are moved across complex 4. So as I said a moment ago, there is a complex 2. Complex 1 interacts with NADH. Complex 2 interacts with FADH2, that new species of reducing equivalent we talked about a few slides back. And so what's key to note is that when FADH2 interacts with complex 2 here, as you can see, there are no hydrogen ions moved across there. So it is a less efficient reducing equivalent, which also explains why we have way more NADH than FADH2. It's just more evolutionarily advantageous to have NADH as the more abundant reducing equivalent as opposed to FADH2. So we have no hydrogen ions moved across at complex 2. We still have 4 moved across at complex 3 and 2 moved across at complex 4. And so we have 4 fewer hydrogen ions that move across the membrane in response to FADH2 when we compare it to NADH. Yes. Sorry, can you say that again?
FADH2 is what donates the electrons to complex 2.
Yes.
And it doesn't come like on the other complex. It comes from the
Correct.
Yes. How does that affect?
The way that that affects ATP generation is that there will be less ATP generated in response to an FADH2 reducing equivalent compared to NADH.
Why does it come?
Sorry?
Why?
Why is there less?
Yeah.
So if we take inventory in response to NADH, at complex 1, there are 4 hydrogen ions. At complex 3, if you combine with the chromosome Q, there's 4 at complex 3 and 2 at complex 4. So that's 10 total. Whereas here, nothing at complex 2, 4 here, and 2 here. So it's 10 hydrogen ions versus 6. So what ends up happening is I'm jumping ahead and skipping myself a little bit. What ends up happening at this ATP synthase, this ATP pump, for every 3 hydrogen ions that are moved across this pump, 1 ATP molecule is generated. So what that means is if there's only 6 hydrogen ions pumped in response to FADH2, we're only generating 2 ATP from FADH2. Whereas with NADH, we have 10 hydrogen ions pumped across. So we can generate more. And so it's a more efficient reducing equivalent, which when we think about kind of like the evolutionary pressures that explain some of the things that happen in cellular and molecular biology. I'm going to go back over here for the microphone. When we think about why things happen a certain way, if we think about the efficiency here, NADH is a much more efficient reducing equivalent, which perhaps explains why we generate more NADH as opposed to more FADH2. Does that help answer your question? So again, the inventory here is 10 hydrogen ions are moved across in response to NADH and only 6 in response to NADH2 because complex 1 moves 4 across in response to NADH. Complex 2, which is specific to FADH2, moves 0 hydrogen ions across. We still get hydrogen ions moved across at complex 3 and complex 4. That's common to both of these reducing equivalents. But because of what happens at complex 2, NADH moves more across. And finally, I said this in response to the previous question, but ATP synthase is this pump that is almost like a gear, whereby when we move across this stalk where we have these complex of F1, F0 in the stalk protein, what ends up happening is that as 3 hydrogen ions move across this enzyme, the stalk rotates about 120 degrees and promotes the synthesis of 1 ATP for every 3 hydrogen ions that move across. And so this is why those reducing equivalents become important, because they help drive hydrogen ions into that intermembrane space, and the accumulation of those hydrogen ions are used here by this ATP synthase to produce ATP, 1 ATP for every 3 hydrogen ions that move across it. So summarizing oxidative phosphorylation, based on the complex proteins on the mitochondrial membrane, for every NADH, we get 10 hydrogen ions that are moved across into the intermembrane space. For every FADH2, we only get 6 hydrogen ions. And again, that's because complex 2 moves no hydrogen ions. And complex 2 is specific to FADH2. And again, we generate 1 ATP for every 3 hydrogen ions that are moved across that ATP synthase. So what this means in total, for every glucose molecule, we have 10 NADHs. And from those NADHs, from those 10 NADHs, we can produce about 30 ATP. If it's 10 hydrogen ions for every NADH, and if we have 10 NADHs total, we generate about 100 hydrogen ions that are moved across. And what I should note is that this is not a perfect system. There is always going to be some electron leakage, and so this will not be a mathematically or computationally perfect process. So it's about 30 ATP from these 10 NADHs. And for our 2 FADH2s that are produced for each glucose, we're going to produce 4 ATP. And so the complete overview of glycolysis of the TCA cycle and from OXPHOS, we get 2 ATP during glycolysis from that one glucose molecule. We're going to get 2 ATP during the TCA cycle. We're going to get about 30 ATP during oxidative phosphorylation from our 10 NADHs, and we're going to get 4 ATP during oxidative phosphorylation from our FADH2s. What that leads to is 38 ATP generated per glucose across all of these metabolic processes that we just talked about. But again, 38 is the theoretical maximum yield here. Again, there's going to be a degree of electron leakage during oxidative phosphorylation. And so as these electrons are being passed across the mitochondrial membrane, it's not going to be a perfect system where we're not going to be able to generate 38 ATP every time from each single molecule of glucose. But down below, we kind of have a summary of what ends up happening where from one glucose molecule highlighted in blue to the left, we're going to generate 6 carbon dioxide, 3 from each pyruvate during the TCA cycle, and 38 ATP. I'm going to stop here and give space for any questions. Yes. Complex 1, complex 2, complex. Yeah, you absolutely do need to know those. So those are proteins on the inner membrane of our mitochondria. What they're important for is with our reducing equivalents, NADH and FADH2, so far up to before today, those are reducing equivalents during some oxidation event. They are what receive and accept the electrons that are lost during some oxidation event. Now, once we've gone to oxidative phosphorylation, they are the ones losing the electrons during an oxidation event now. So they are losing the electrons that are going to be passed into a series of molecules that carry them across the membrane of our mitochondria. Why that's important is because these protein complexes 1 through 4, they're channels that open and close, and what regulates their open and closing is the moving of electrons across from complex 1 to 3 to 4, or 2 to 3 to 4. And them opening and closing by nature of these electrons moving across them, carried by iron sulfate, coenzyme Q, cytochrome proteins. The opening and closing of these protein complexes allows hydrogen to move across the membrane into this intermembrane space. That moving of the hydrogen ions are key because that's going to be used by ATP synthase to generate ATP for every 3 hydrogen ions. Does that help, like putting it together like that? Does that help?
I guess like my question would be like what extent of detail, like you need to know like coenzyme Q for like 1, 2 to 30, or is it more just like the general picture of what?
Yeah, you need to know those details. Knowing what which reducing equivalent complex 1 interacts with, which reducing equivalent complex 2 interacts with, how many hydrogen ions are moved across each protein complex. Yes, absolutely, you need to know that. Yeah.
So when ATP 5%.
Yeah. Yeah. In the next slide, we're going to talk about what happens to the ATP. Yeah. I like where your head's at. Any other questions? All right. So we have our ATP. What happens to it? So in the inner membrane of our mitochondria, there are what's key to note is that inner membrane is impermeable to a variety of molecules that are particularly polar. And again, that's meant to try and protect our mitochondria from only enabling what's supposed to be there from being present. Kind of like what we were talking about with the nucleus, where there are really vital functions that are happening in this organelle. So it's important to have a careful regulation of what is allowed in and what's transported out. And so we have this transporter that will help move ATP out and move ADP in. Excuse me. To enable that ADP to help generate more ATP. Excuse me. And to help shuttle out the ATP that's been generated. In addition to that, we have transporters that help shuttle pyruvate in, because again, glycolysis happens in the cytoplasm of our cells. That pyruvate needs to make its way in, bless you, that pyruvate needs to make its way into the mitochondrial matrix where the enzymes for the TCA cycle are present. In addition to that, the series of intermediates during the TCA cycle, bless you, it's like 5 sneezes, just like right off the bat. Sorry, it's like I feel like I should like coordinate and conduct y'all sneezing. So there's a lot of intermediates during the TCA cycle, but there are a few of them that are key that allow us to plug into the TCA cycle from sources of energy that are not glucose. And so what that looks like is we can use amino acids to feed into the TCA cycle. As an example, again, during either a period of starvation or muscle wasting, muscle will be broken down into its constitutive parts, the amino acids, and a few specific amino acids can feed into the TCA cycle. Via a transamination event, alanine can become pyruvate. During another transamination event, aspartate can become oxaloacetate. And finally, glutamate via transamination event as well can become alpha ketoglutarate. And so this enables the ability of our cells via these transamination events, in the absence of glucose, to take amino acids from protein and have them plug into the TCA cycle. And so this actually permits both the breakdown, the catabolism of muscle, or the anabolism, the synthesis of amino acids that can be generated and used to synthesize protein. In addition to amino acids being used to plug into these processes, we can also have fats or fatty acids be used to plug into these processes as well. That's mainly via a specific oxidation event, a beta oxidation that allows coenzyme A, we talked about with acetyl-CoA, coenzyme A to be linked to a carboxyl group on some specific fatty acids. And so on a fatty acid, we'll have acyl-CoA dehydrogenase to enable the production via this dehydrogenase event and a second dehydrogenation event that leads to an oxidation event on a beta carbon, beta oxidation, to eventually produce acetyl-CoA from our fatty acids. And so the breakdown of fat into fatty acids allows us to use them as a source of energy to produce acetyl-CoA that then feeds into the TCA cycle. So it can be used as a substrate for energy. These, as is the case with a lot of these reactions, they can work in both directions. So what that means is in the abundance of glucose that produces acetyl-CoA, we can go from acetyl-CoA to fatty acids that leads to the synthesis of fat tissue or lipids that then leads to fat tissue. And so another key thing to note is that this also produces a reducing equivalent, FADH2 and NADH in the middle. And so this produces both acetyl-CoA and more reducing equivalents that feed into oxidative phosphorylation. And so the precursor for these fatty acids that we looked at in this previous slide is usually stored as triglycerides. When these triglycerides are broken down, they produce both glycerol and fatty acids. The fatty acids, again, can via beta oxidation, can produce acetyl-CoA. And the glycerol backbone here on these triglycerides can be used to produce glyceraldehyde 3-phosphate that can feed into glycolysis or gluconeogenesis. And so the triglycerides that we have in our bloodstream can be used both to produce glycerol that feeds into glycolysis or gluconeogenesis and the fatty acids that can feed into the TCA cycle. And so this snapshot helps us understand how fatty acids can be used as a substrate or an energy source to feed into these different metabolic processes that we've talked about. I'll stop there and give space for more questions. Yes. I love those. Yep. Mm-hmm. No, it can be happening simultaneously. So there's I'll direct you to slide 13. You can appreciate that all throughout these complex proteins are there are many of them across the inner membrane of our mitochondria. And so at any given moment, there are lots of ATP being generated across many complex 1s and 2s across the surface. And that it's, what's a good way to say this? Whenever we talk about these proteins that we illustrate on either the surface of a cell or in a cell, there's not just one of them. There are typically thousands or tens of thousands. And you don't just have one mitochondria in your cell. While your cell only has one nucleus, there are many mitochondria through your cell. And different cells, based on what their function is, will probably have a lot more than others. Your skeletal muscle will probably have a lot more mitochondria than, say, a smooth muscle cell in your stomach, for example. And so the ability to produce lots of ATP is mediated by having many mitochondria in a single cell and many of those protein complexes across the inner membrane of a single mitochondria. Does that help make sense? Okay. All right. If there are no more questions, I think that is our last slide for today. So I will see you guys on Tuesday. Hope you guys have a good weekend. And I'll stay behind if you have any more questions.